Okay, today what I wanted to do was go over a simple analysis to look at good data and bad data within the field breeding program. So what I'm showing you here is our standard master note sheet. That's the notes that are collected by the end of the year. We have a barcode. Each one of these is specific to an individual plot, that is a set of plants within a designated location within the field. That's broken down further into the location, which is a combination of the actual physical location plus the year. The year is in here. The test, which usually is a block of things that are all tested together. The plot number, which always starts at 1 and goes up sequentially up to the maximum number of plots. And you'll see that this plot number is part of the barcode. We usually have an entry number and a replicate number, which aren't particularly filled in this particular example. We have a stock number, and in this case, this is what the seed can be traced back to. This is part of the Genomes to Fields project, and so we were sent this seed, so these stock numbers are not our traditional stock numbers, which look more like a barcode. There's also a pedigree, which is the genetic information that's involved in this. And then we have a number of different measurements that we've taken. For instance, days to anthesis, which is calculated by subtracting the date that the seed was planted from the date of anthesis and in the case of days to silking, from the date of silking, which was taken at 50% of flowering. We have PHT, which is plant height, FHT, which is flag leaf height, EHT, which is ear height, STD, which is stand count. We have a spot for lodging. And then once we combine harvest, we have a spot for the plot weight, or the actual grams of seed that are harvested within the 25-foot plot. We have a moisture, that's the percentage of water in the sample that's harvested by the combine. And then we have a test weight, which is akin to the density of the grain. We have columns here for calculating the yield in terms of tons per hectare, which is the international scientific unit, and then corrected for moisture, because if you imagine somebody would not want to compare a sample that's 50% water to a sample that's 10% water. So we calculate at 15% moisture on average. And then we also have bushels per acre, which is also adjusted for the moisture. We have a plant population, which is the size of the plot multiplied by the stand count, so that we get on average roughly 30,000 plants per acre. The seed that was planted, which field it was in, the number that it was harvested with the combine, and then very importantly, the range and row number. And these are spatial coordinates in the field. So given some field, what position in XY coordinates is that particular plot? And I'll show you when we go through analysis that this becomes important. We also have a number of other spots here for additional data, and any other data that's collected, we just put at the end here. The reason we try and maintain a consistent format of these yield trial spreadsheets is so that at the end of the season we can combine them all and run a comprehensive analysis, or we can combine multiple years. So we stuck with this spreadsheet format maybe 5 or 6 years ago, and I don't anticipate changing it anytime soon. The next thing I wanted to show is data analysis through JMP. So the way that we get the data into JMP is once we have that spreadsheet that's completed from our master note sheet, we just simply go to open. Once we've opened JMP, we can select it, and then it's going to give you a number of different tabs that you have an opportunity to analyze. For the case of this one example, we're going to look at CS20, G2, F2, and we're just going to import this. We might as well just leave every other setting as default for the moment. Now, the first thing I really want to show you is how to screen for outliers. Outliers are very important because they're going to ruin all the statistical analysis. And at this point, before we've harvested, we still have an opportunity to go back into the field and harvest or measure anything that we missed measuring the first point. So assuming that this data imported correctly, which we'll find out very quickly, what I like to do is I like to go to Analyze. We could look at the distribution, and that would show us if there's any extreme outliers. But I like this fit Y by X. And what I like to do in this fit Y by X is select all traits and turn them into both Y and X factors. Now this gets a little overwhelming. It might be too much to show in the video, but if you have time and you're working through this data yourself, it's a good way to do it. So what you're going to see in this case is a scatter plot. And for here, in this particular cell, we have days to anthesis by days to anthesis. And then over here, we have days to silk by days to anthesis. Now, what we can see already is that there's 1 point, it looks like, at 0. This is actually most of our points. And then we have another point down here at negative 45,000. Now, that's absurd. So what we're going to do is we're going to click on this so that all the points get grayed out. And then we're going to simply draw a box to the 1 point that we want to investigate. Now, this is why I like JMP. I'm going to put this window over to the side, and we're going to go back to our JMP spreadsheet here. And what you hopefully will see as I scroll down, I'm on the right tab, is a number of points that have been highlighted. And if we look at what that measurement was, days to anthesis by days to anthesis, we can look in this column right here and see that we have a number of negative 43,901 points. Now, this comes from subtracting the planting date from the date to anthesis that was measured. If you look over here to this column, those dates were not actually recorded. Those notes got missed. And so when we subtracted the planting date from that and then turned it into a numerical value, funky things happened. So what I really like about JMP is I can now come in and I can just merely delete these points. The problem with doing it in this way is, first of all, in this silly example, I have way too many points to do this for. This works better if you just have 1 or 2 outliers. The other thing I really like about doing, the other potential problem about this is next time you go to open your Microsoft Excel spreadsheet and import it into JMP, those observations that you just hid in JMP are still there. So we really need to go through and delete these in Microsoft Excel. So I'm going to go ahead and do that and pause the video and come back in just a second. Okay, so I just deleted those points. One thing I always recommend is you continue to save things as new versions. The memory is cheap, and if you do make a mistake, it'll be really easy to go back to an old version and recover it. And I just keep saving them as version 2, version 3, version 4. Sometimes we get up into 80 or so versions, and that's just fine because, again, memory is cheap. So I save that. Now my next thing to do is to go back to JMP and import that new Microsoft Excel file. So it's 2, and luckily this is still highlighted, so we can just say import, and we're right back where we started. We're going to go to analyze, fit y by x, select all of these things that were measured, and go ahead and do this again. And now what you can see, this was our original in this case here, and we only have these 2 points because this negative 43,000 really skewed our viewing. So after we delete them and then replot them, you can see that now it ranges from 60 to 76 instead of from negative 43,000 to 76. Okay? So that's the way I clean the really extreme outliers. But now what we're going to go in and do is find some of the more subtle outliers. So a good example I see right here. So this point shows that our plant height is less than 10. If you see the bulk of the other points here, it's very clear that that was an error that somebody had recorded. So I'm going to go back to my JMP spreadsheet. I'm going to find that selected cell, and I am going to either delete it, so we can see it's an 8, so we can delete that. You also have an ability to fill in a color to this cell if you just want to know where to go back and delete it in your Microsoft Excel spreadsheet. So you can see I deleted it, but you can see it's still here. So if you really actually want to get rid of it and replot your graph, you're going to have to go back and reanalyze this. Select all these again and put them in my Y and my X, and we'll plot them. Like I said, if for the purposes of video, we probably should have gone individual by individual. Now, when we get to something like this, this isn't really clearly an outlier. Well, it is an outlier, but it's not really clear that it's a mistake, because that seems to be a reasonable days to silk. And if we look at flag leaf height, it's certainly within the realm of possibility that you could have a very short plant. But the flag leaf height has to be shorter than the tip of tassel or plant height, right? So we can go and we can look at this, this plot here, plant height by flag leaf height, and we can see indeed the plant height is 75, the flag leaf height is 30. Seems really sketchy, but it's not technically an outlier. Everything that we know to be true is still true about this. Same thing with this point here, or actually not the same thing with this point here. This is at 60, and here we have a flag leaf height that's taller than the plant height. Chances are what happened is when the students were entering this data, they switched flag leaf height with plant height, and that this should actually be 77 for plant height and should be 60 for flag leaf height. So that would be a relatively simple change to make. This is another very clear outlier. So you can't have an ear height in corn that is 350 inches in this case. We're measuring inches, not centimeters. Probably bad for our international audience, but it's just the way that things are reported usually here in the United States. So both these 2 points are clear problems, and I'm going to go and I'm going to delete them. And I'm going to highlight them back in red. Actually, in this case, oops, do that. In this case, it was the ear height. That's wrong, because the ear height is actually taller than the plant height. So we know that's a problem, right? I'm going to highlight these two again. And if you hold down Control, you can select multiple points at the same time when you draw your box. So I'm going to find that other point that's highlighted. Here we go, 335. So that's obviously wrong. I need to make sure I color in that cell so I know to remove it from my Microsoft Excel spreadsheet. And I screwed up that other one by not doing the same thing too. So we need to go back and, okay. So that's how I go and delete outliers. There are clearly other ways to do it. I used to do things in R, but this interface of JMP where I can very quickly plot anything I want from traits that were measured in the field and visualize them and be able to select things that I know are wrong is very helpful. Now, when we go to do our statistical analysis, we'll find some other outlier points that we need to get rid of. For instance, I'm not going to delete this one. I'm pretty sure our statistical analysis will find this point right here. Okay, so this is a process. It's not something you do all at once. And for most trials, it's probably going to take an hour or so to clean up the data. Okay, now what we're going to do is we're going to analyze this data to look at variance components. And what variance components mean is where the sources of variation in our experiment are coming from. Now, what we hope in most experiments is it's coming from the genotype or the pedigree. What we often find is there's other sources of variation. So just to start simply here, I'm only looking at one test. This is the G2F dryland test. And I'm going to go here to Analyze, and I'm going to go to Fit a Model. Now, this model is going to be very simple. It's going to, we're going to look at all the traits. So in that case, it'll be a little more complicated. But what we're going to use as predictor variables for this are the genotype and then the row and range effect. Now, what the row and range effect are spatial positions in the field. And the reason that these are significant is because we use furrow irrigation. So that means there is a hill that the corn is planted on, and then between the hills there are furrows, and down that furrow is where water runs. Additionally, we use fertilizer that runs down the row. And so what we see is different rows have happened to get different amounts of water and different amounts of fertilizer. But additionally, over these fields that have been set up for 50 years, when we're always furrow irrigating and adding water to the front of the field and it goes to the back because there's a slope, we see it washes a lot of those fine clay particles to the back of the field. And that makes the back of the field a heavier soil. And we see some stunting of the plants in the back of the field relative to the front of the field. So for those reasons, we usually see variation from both the row and range spatial effects. Now, I don't want to put these in the model yet because they're actually listed as continuous variation. So what does continuous variation mean? it means we expect that range 1 is going to be either greater or less than range 2, and then that same order will continue on to the end of the field. But we don't believe that. We believe range 1 might be high, range 2 might be low, range 3 might be high, range 4 might be high, range 5 might be low, et cetera, et cetera. And so we actually need to click on these, and it's very important you click right on this blue part and change them from continuous to nominal. So these nominal, categorizations will make it so that it's appropriately analyzed. So I have my traits: days to anthesis, days to silk, plant height, flag leaf height, ear height, and stand count. I now have the predictor variables that I think are going to condition these. But there's one other thing we need to do to get variance components, and that is we need to select all of these predictor variables. So I'm holding down Control and clicking on each of them. I'm going to go to Attributes, and I'm going to turn them into Random Effects. These random effects will use a restricted maximum likelihood model. This checkbox, there's various reasons to keep it checked or unchecked. I'm going to keep it checked for now, but our model is now set up to run. I'm going to keep this dialog box open so I don't have to go through this whole process to fill it out again, and hit run. It's a very simple model. It's a simple data set, so it worked very quickly. So what do I find from this? What I find from this is we have a model for days to anthesis now with pedigree, our range and row spatial effects, and residual, which means variation that we could not actually attribute to anything. So when I look at this, I see that 49% of the total variation for days to anthesis came from the genetics. So that's good. It's not great for days to anthesis. There's reasons for that I won't explain now. We see that 10% of the variation came from the range. So that's from the front of the field to the back of the field. We see a 10% of the total variation came from that. 4% came from row, and 36% of the total variation is unexplainable. I can click down on this effect details here, and I can actually look at what the best estimates in this model are for each of these, for when they had flowered for days to anthesis. Okay, so that's useful. Click this back off. I can also go to my random effect predictions, and so this will tell me, for instance, which ranges and rows had the largest effect. So if we look at, for instance, in range 2, it said it flowered 1.3 days earlier than the average of the test, essentially. And then if we look at range 15, it says it flowered 1 day later. Okay, so this is enough variation here, having a difference from 1.3 to 1 day, so that's a 2.3 day difference, to actually call this term quite impactful, to explain 10% of our variation. If we look at our row variation, now there's a lot more rows to spread the variation between, but we're talking about a half day of flowering time. Right? So nothing too extreme here. So if we took the extremes, minus 0.7 to plus 0.65, so that's like a 1.3 difference in flowering between row 13 and row 69. Okay, so there is a difference there, but it's not as big as the difference between the ranges. The last thing I'm going to show in this section is to talk a little bit about the second last thing. Talk a little bit about the model. So we're going to go back up here to days to anthesis. The R squared of this model is 0.8. R squareds are not usually that useful of a measure in this case, but it does tell me that this is an acceptable model. It tells me how many observations are here. So if I only saw there were 2 observations, then I know there's something wrong about how I put my data in. In the case of this G2F trial, there's 550 plots, and some of those plots were missing data. So that's why we see 534. There were more missing data in days to silk than there were in days to anthesis. That's why we see 524 here. Okay. Now the other thing, and I think this is the last thing I want to show in this section, is we can actually look and get connecting letter reports on which varieties were the earliest to flower. And this is another good way of troubleshooting. So I'm going to go to the LS mean students t-test. I'm going to select this. You're going to get this very confusing matrix that you can change in your default to not show, but you can also just click on this little red box here and eliminate it. And here's our connecting letter report. So the way to read this and understand this is this hybrid here, TX773 by TX779, has the letter A. The only other two hybrids with the letter A are these two here. So this hybrid is significantly different from everything else in this test other than these two. Okay? All three of these are from the Texas A&M corn program. I would expect that this would be the latest hybrid. And when we look at what the estimate is, it flowered in 73.85 days. It's clear that that's by far the latest. As we scroll down to the other extreme, we can see CG44 by CGR01, CG119 by CG108. These are both lines or hybrids from the Guelph, Canada program. They have their own connecting letters, so they are significantly different from everything else. They flowered in 62 to 64 days approximately. So that was, I believe, 9 days less than our latest flowering material. Okay? So if you see anything here that just looks really odd, it's another indicator that you might have an outlier. For instance, one observation here could be 73 days. The other observation could be 95 days. And so then it would make things look a little weird. So I'm going to end there. So this is how you do random effects, variance component estimates. And I will switch over to fixed effects in the next video. Although I realize there's one other thing we should probably do, which is compare the variance component effects between these. Okay. So when you run a random effects model, you get these variance components. And the variance components are presented here. And so we can see that the pedigree or the genotype or genetics has a variance component of 2. The range has a variance component of 0.45. The row has a variance component of 0.18. And the residual or the random unexplainable error has a variance component of 1.5. Now, one of the ways I like to look at this, because these numbers are sort of meaningless, is the percent of total. So if we sum up all of these variance components, we get this total. And then we can divide each of these into that and get the percent. And so what this tells me is 49% of the variation for days to anthesis or DTA, 49% of the variation comes from pedigree, 10.7% comes from range, 4.3% comes from row, and 36% comes from unexplainable error. That unexplainable error comes probably from people either not doing a great job of recording that date, recording the date incorrectly as they entered it, or more likely a seed mix-up that occurred sometime between making the spreadsheet of what should go where and pulling the seed and actually putting it in the ground. So all those things can contribute to the residual variation. And we can see for something that's somewhat subjective like days to anthesis, that person to person, the residual variation might be more or less there. If we look at days to silking, which is when the silks come out of the ear, we see that there's more unexplained residual variation. We see the numbers between range and row are about the same, and that's because that spatial variation caused about the same delay in flowering. The reason I think we see more residual error here is due to human error. The silks come out within the canopy, so they're often covered up by leaves, whereas the tassels, when they start shedding pollen, is something that's very easy to visually see. So we tend to see more residual error in days to silk. Let's look at plant height. So plant height is typically our most heritable measurement, meaning it should have the highest proportion of variation due to genetics. We still see that range and row are significant and substantial, but we also see a very, very high residual. So this tells me that there's probably some issues with our random effects estimates, or with our measurements, random effects measurements of plant height. If we go and look at flag leaf height, it's even worse, so not a very good job there. And if we go and look at ear height, this is actually quite strange. Ear height is usually the worst of all of the measurements, and in this case, the amount of variation due to genetics was actually fairly high. Our stand counts, or the number of plants that, that emerged and are actively growing, is usually not a very genetically dependent trait. And we see 12% of the variation came from there, a little spatial variation, and then the unexplained variation is very high. So that's a rundown of all of our traits there. And so now we can move into fixed effects analysis. Okay, for fixed effects analysis, we would never have turned these into random effects, or we can just simply turn them back off. So I'm going to select all these by holding Control. I'm going to go to Attributes and click off that. And now we're back to the standard least squares model. Now there's two reasons that you might want to do this. First of all, you might want to see if the effects of these are significant. So does genetics actually significantly affect these traits? The other one, and the more important one at this moment, is to look at the impact that these have on outliers and creating outliers. So we're going to fit these separately. There's no reason to fit those together. And your jump may have a little bit different output. One of the things I'm looking for is the row diagnostics tab here. So we click on this little red box here for the trait we're interested. We go to row diagnostics, and we can either fit residual by predicted or actual by predicted. I like residual by predicted. Okay, so I'm going to make this figure a little bit bigger. And what you're going to see is this striping. The reason for this striping is because we have something that's not actually continuous variation. So what that means is there's no way we would have given a score to something that it flowered in 66.5 days. It either flowered at 66 days or it flowered at 67 days. And so because of that, we see these stripes are approximately 1 day apart. So on the x-axis here is the day that was predicted based on our model, and our model includes genetics, the range variation, and the row variation. And then over here, we see the residual, okay? And so it's taking the two replications that we have of every plot, and it's fitting this model to be able to identify where things don't match up. So in general, I would say I don't see a lot of major outliers in this particular trait. We did clean up all those extreme measurements. If we look here at our effects test, we can see that pedigree is significant. There were 250, as we lose 1 degree of freedom, 250 different genetic pedigrees. There were 14 plus 1, so 15 ranges, and 36 plus 1, so 37 rows of plants. So row is less significant than range. Just like in random effects, we could go and look at the connecting letter report, and what you're going to see is the fixed effects is less conservative. So now it estimates that top line or top hybrid at 76 days of flowering, and the bottom hybrid at 61 days of flowering, as opposed to the random effects model, which compresses all the variation to the mean. So you don't see that quite that extreme. This leverage plot is really helpful too. So we're looking at leverage based on pedigree here. And so I don't see any major outliers. I mean, this point right there might be a little bit concerning, but not enough to have reason to delete it. We can look at the range. Is there any range variation that is concerning, or row variation? So overall, I think this data looks good. It doesn't look great because there wasn't a lot of variance explained compared to what I was hoping for, but it's decent data. Plant height is something that actually is measured much more quantitatively. So we measure in inches, and so we would expect when we go to look at our row diagnostics fitting residual by predicted, we see that there's not those stripes that we saw on Day's Anthesis. And as I zoom in here, I don't see anything that's a clear outlier. These short plants here, you see that they are very close to the center line. So their residual, that is the unexplained variation, is very small. The reason these are so short, right, because this is inches, is because they're probably from Guelph, Ontario. That material is just unadapted. The Canadian material is unadapted to our Texas environment, and so it flowers, or it's very short. So here we go. CG 119 by 108, so that's a Guelph line. CG, CG, so yeah, those are Canadian material. And then if we look over here, I would guess it's our Texas lines again. Not as likely to be true as the short material, so that's not a Texas line, that's not a Texas line, not a Texas line, not a Texas line. Okay, so I guess some of the Midwestern material is where we're getting most of that height from. And that might change from year to year. In this case, rho was not significant. Okay, so that means there was variation that is significant, like the differences are clearly caused, or we can't say that they're not caused by genetics or range, whereas with rho, it's not significant. So technically, we should probably delete it from our model. So what would that mean? It would mean coming in here, hitting delete, and hitting rerun again. And now we can compare this model, which I just deleted range, which I just deleted range for, okay, with this model. So we'll go back to plant height. That height is there. And we can see now our model pedigree, range, rho. Rho was not significant, we deleted it. And so we now have pedigree and range. What we can also see is that the that the fit of the model would have changed. So our R squared dropped a little bit, but we dropped the non-significant term. And we would also see that our connecting letter reports are a little different now. Okay, getting back at it. What I just did was stack the genomes to fields dryland trial along with the genomes to fields late planted irrigated trial. So not the best two to stack, but unfortunately our genomes to fields irrigated trial, the notes are not available yet. So I stacked these. That's one of the nice parts about our spreadsheets all being in the same format, so I can line them up fairly easily. And I already got rid of the outliers in JMP. So it's imported into JMP, so I already showed you how to do that. The thing I wanted to show you here is it's really nice to have the genomes to fields trial where there's 2 replicates of every genotype or every hybrid. So that allows us to compare replicate 1 to replicate 2 and see where there might be errors. But when we have the genomes to fields trial with all 3 variants, that means we technically have 6 replicates. Now, each of the G2F trials, 1, 2, and 3, are subjected to different conditions. So the first condition is optimal. G2F1 is optimal, best fertilizer, irrigation, whatever it needs. G2F2 is dry land. So that means we cut the fertilizer typically in about half, and then we also never irrigate it. G2F3 is mostly optimal, so optimal fertilizer, optimal irrigation, but then we delay plant it approximately a month with the hope that that's going to stress the plants out more and be more reflective of maybe what a planting might be under a climate change scenario or under some type of extreme environmental stress. So in the ideal situation, every genotype would be replicated 6 times. In this case, we can go to, I don't sort very often in JMP, but we could sort, for instance, by pedigree. And it opened a new window. And what you can see now is in the case of this hybrid here, which is one of the so-called yellow stripe hybrids, so it's replicated multiple times, we actually have 1, 2, 3, 4, 5, 6, 7, 8 replicates of it. Now, unfortunately, a lot of the data is missing, so we still need to get that collected where we can. So these are all yellow stripes, but as we get into the meat of the test here, we should only see 4 replicates. And so in this case of this hybrid, we have 4 replicates of this, of plant height, and flowering, flag leaf height, ear height, etc. All right, so now what we're going to do is we're going to analyze this. What I wanted to show you is taking it to the next step of how we analyze multiple tests together. So we put in the genotype. We're going to remember to turn these into nominal traits, range and row. But range 5, for instance, in G2F1 does not necessarily have the same effect as range 1 or range 5 in G2F2. So we need to put in this test. And this test is going to separate the G2Fs along with some special yellow stripe hybrid trials that are probably too complicated to get into here. So I'm going to add that in here. And then what we're going to do is we're going to take range and row. We're going to add those in, but we're going to actually nest them in test. And so what that means is that range 5 in G2F2 or range 5 in G2F3. Okay. We're going to take all the traits we looked at before. We're going to keep this dialog box open. And then if you remember, we're going to turn these all into random effects so that we can look at the variance components. I'm going to click this box off because it's going to help not give errors. It might take a little bit longer to fit. This is quite a bit more of a complicated model to fit. So you saw it might have taken 10 seconds instead of 5 seconds. But we've got our answers right here. So what does this tell me? So for days to anthesis, or how many days it took from the time we planted seed to the time 50% of the plants in that plot were flowering, 43% were due to genetics or pedigree, 12% was due to the test. So there was a difference between the early planted and the late planted G2F. 8% was due to the range variation, and 7% was due to the row variation. And we can go back up here into random effect predictions and figure out exactly which range it was which range and which test that was giving us a significant estimate. And we should see something similar to when we just analyzed G2F2, G2F3 in this case. Let's go to plant height. All right, now this is extremely unfortunate. Only 15% of our variation is due to genetics. 58% of our variation is due to the test. Actually, it's not unfortunate because what that means is the late planted plants grew either taller or shorter than the early planted plants. We still see that range and row are a significant amount of, or a substantial amount of variation, and then we have this effect from the residual. So when we actually go and look at this, it looks like the test, the average plant height in G2F3 was 94.7 inches. The average height in G2F2 was 80 inches. And so that's probably a difference of planting date plus some types of irrigation or fertilization. We can go and we can look at plant height again in the same way we did before with the student t-test. Go ahead and click off that crosstab report. And now we see there's much less of a significant difference in this two-test plant height than there was for days to anthesis. We see the tallest plant was 97 inches, and then the shortest plant, the estimate is 66 inches, and that's, again, the Guelph, Ontario lines. Okay, so that gives you an idea of how we might look at this data, and I'm going to end this video here, probably get some feedback and figure out what else and answer people's questions.